Everyone again here. Today I'm going to show you something that it has nothing to do with mosquito. So I don't know honestly how I ended up in this session, but may when you know people in the area. So what we're going to talk today is something about a particular molecule from black flies, and this is more like a discovery-driven hypothesis we follow in the lab rather than, discovery rather than hypothesis-driven, and it has to be with the mining drugs from blood feeders. So just really quick, and how we started with this one, background on what we have in this SIGPing, and I'll let you know, in a little bit, I'll tell you what we call that molecule SIGPing. So SIGPing belongs to the D7 family of protein from the blood-feeding Nematocera. It's a short form. In the black flies, they also have the long and the short, just like in the mosquitoes. It's right here. SIGPing is right here, and this right here. And one of the other reasons, if you look in this in clay here, this is right here, they are about 60 to 70% identical in the three species that we have sequenced in the transcriptome. And one of the reasons I was interested in particular in this in SIGPing, the SIG-128, it was because of this molecule here. Simulidine wasn't published and described as a thrombin inhibitor in Simulium vidatum, and that is something that we haven't seen in D7 proteins. So I was interested more in the mechanism of action, and so that's the reason we picked this molecule from Simulium nigrimano. So we express the recombinant protein in E. coli, soluble fraction. There is no tags or anything, so we had to send everything after the purification out for Raman degradation and mass spectrometry, just to make sure that everything that we have here is the protein. We ended up with a really pure protein, good quality, and we had to LPS decontaminate because it's coming from the soluble fraction. Now that we have the protein, the recombinant protein, ready for our experiment, the first thing that we did was, of course, since it was annotated as a thrombin inhibitor, we went ahead and we did the enzymatic assay, not only thrombin, but another 16 proteases that are involved somehow in inflammation, platelet aggregation, and coagulation, and we see that there is no activity at all. Same thing, since we know that it belongs to the D7 and our previous experiences, we know that D7s are a scavenger of small molecules and lipids. We run the calorimetry with everything we have at hand, and we were all negative. There was no binding detected. Same thing for the classical assays that we run in the laboratory for blood feeding or arthropods and everything that comes from salivary glands specific from females. And we didn't see any effect on coagulation or platelet aggregation or anything. So we went to go back. We were either the protein is in really bad shape, which I doubt at the time. We know by CD spec and everything that it was in really good shape. So we went back and we looked to what is published or what was published in the literature in using salivary gland homogenate of black flies and looking into something else besides what we usually do, coagulation, aggregation, or vasoconstriction. So what we found here is the black fly Simulium vidatum has at least the salivary gland homogenate has a strong inhibition of proliferation of B and T cells. So with this in mind, we went ahead and we put all the recombinant proteins from Simulium, Guianense and Simulium nigrimanum that we have at hand. And what we can see here, the only one that was positive for inhibition of T and B cells, this is one is made out of the splenocyte. It has all kind of things inside, from B, T cells and everything that you can find in the spleen cells. So we, just to mention here, this is SIM25 and SIM3. They are the other members from the D7 from the same mosquito species. One is the long form, the SIM3 is the long form, and the SIM25 is the short form. So it seems like it's specific. So we named this protein SIGPing after Simulium cell proliferation inhibitor. So we repeat the experiments with using the human lymphocyte and all the different preps, and we can different concentration, and we see that it works really well inhibiting proliferation of the spleen cells. And with the mitogen we used was concanavalin 8, so having this in mind, and the D7 that is scavenger of protein and has a really nice pocket in the middle of the core, we run the calorimetry with the concanavalin A, and there is no binding either. So if this molecule is inhibiting EU, which is inhibiting proliferation of T and B cells, is doing by another mechanism rather than removing the mitogen out of the equation. And we also tried different mitogen, the one that everybody uses in the field. So CON8, LPS, and CD3, CD28, and POKI-WIT, and all of them work just fine. So that led us to think that it's definitely doing something else, the mechanism of action. So what we have here is by flow cytometry, just to, as I mentioned before, the first experiment were kind of raw with the whole splenocytes. And this one, we are separated T and B cells, the B cells by negative selection, B cells and T cells here. And what you can see here, this is when you put the CON8 or LPS, you can see this is the inhibits really well the cell division. What you measure here is like a cell division. CFSC is a dye that goes in, permeate in the membrane that, excuse me, membrane in the cells. So every time they have a division, they have, and that's the reason you can see here. When you have a sick pin present, there is no cell division. And the same thing goes for the colorimetric assay, using the same subset as in T, the B and T cells, and so we know that sick pin both inhibit both of them, so either T or B cells. So that's when we went after that, just to see the mechanism of action. So what the first thing that you think when there is an inhibition of T cell proliferation or division is easy, the easy things to think is either by necrosis or apoptosis. So that's what we did in the next experiment. So what we have here, just like as a control, here is the resting cells without and with sick pin. So, you know, just to have a baseline, so it's about 5% necrotic and apoptotic, so there is no difference in the presence or absence of the sick pin. Now, if you stimulate the cells and you do exactly the same experiment, you can see that number for the percentage of apoptotic and necrotic cells are pretty much the same as the control. So we just tell us there is no killing the cells. And we know also there is no killing the cells because we did a different experiment. When you incubate the cells with the sick pin, let it for a little bit, for 20 hours, then you remove and wash the cells and put it to proliferate, and they proliferate again. So the next thing is like, well, maybe it's either a cytokine or a cell receptor in the T or B cells. So for that, we set up a Viacor experiment, and this is a little bit different. So we mobilize a recombinant protein, and we flow over the sensor everything we had that was resembled to stimulants or something that can promote proliferation and activation of the T and B cells. And so you can see here, the only one that is a positive signal is the CD4. And CD4 is a receptor of the T cells. And the difference in the intensity here is just because there are two different companies. One has a fusion tag and the other doesn't have the fusion tag. So the sensogram, the response units in the Viacor is a little bit higher than the other one. CD4 is, for those who don't know, is one of the markers for the HIV patients, the one that GP120 binds and it facilitates the invasion of the cell. So in a more detailed kinetic experiment, we just run again the experiment, but just with a purified system, and you can see here that is a pretty high affinity for the receptor. And the same thing as a proof of concept, just to like orthogonal method, we use an ELISA just to do pretty much the same experiment, and we can see that it's a specific, there is no false positive or anything. So the last experiment that we did was just like see, by now most people say like, oh, well, big deal, you know, this is inhibiting T cell proliferation or B cell proliferation, but what is the relevance for the biology of the fly? Because for the division of the T cells or B cells, it would take 1 day for it to start dividing. But when you have activation of T and B cells, immediately you have releases of different cytokines that can trigger mast cell degranulation. In mast cell degranulation in the bite site is extremely bad news for the feeder because it has release of histamine, leukotrienes, and all kind of things that give you the itch, and they will trigger the awareness of the the host. And another way to look at this is by local inflammation. This is a pal edema formation where you inoculate or co-inoculate the recombinant protein with the stimulus carrageenan. That's the same one that the pharmaceutical industry uses for cream and that kind of things like they put in the animals. And what you can see here, the sigping, either a 5 microgram or 15 strongly inhibits the inflammation. So if you look at the paw, the mouse, it just goes back down really quick. And if you cut the paw and check the cytokine, excuse me, in that tissue, you can see here there is significantly reduced MCP1, IL-6, and TNF-alpha. Especially MCP1 is like a really activator of mast cells. So that's what we think it is doing for the biology of the fly. The last one, this is just like since we are filing a patent on this molecule, one of the things that we were asking was to compare what is in the market. So cyclosporin A is what most people take when they have a tissue transplant to reduce activation and proliferation of T cells. If you can see here, we did experiments side by side, and cyclosporin A works really well in T cells, but doesn't work too well in B cells. And sigping, in comparison, has a better overall activity. So just to conclude here, sigping is a potent splenocyte proliferation inhibitor. It inhibits proliferation by other than killing the cells. Is more like acting directly into the CD4, and that's one of the other things, a possible application to use this as a treatment for HIV invasion. And it also reduces the local inflammatory activity in vivo. And to here, at the time we were done with this, I wasn't convinced that inhibition of T cell proliferation was actually the biological relevance activity for the fly. So we continue studying this molecule, and we found out that it's actually inhibitor of the complement system. And most of them blood feeders have inhibitor of complement system, and this one in particular binds to the C3 convertase, which is the common pathway for the 3 pathways, the alternative, classical, and the lectin. So we are pretty sure that this is the function of the salivary function of this molecule. So with this, I'm done, and I just want to thank the people that have done this one, especially Jose, the boss, and the person who actually did all the work. This is a very long project, along with Fabiano sitting there and Andresa in the back. She did all the work, and for that I'm very thankful. So thank you very much. If you have any questions, I'll be glad to take it. Well, with this sorry, I was concluding the session. Sorry.
I think you said that it also inhibits B cell proliferation.
Yes.
But B cells don't have CD4. So
No.
It's doing something else.
Exactly. We don't know. That's the reason we separate the B and the T cells, because at the beginning was all together, so you never know what it is. We don't know. We were trying, Andresa was doing something with the TCR activation downstream to see the signaling of the cascade, but we're still working on it. So.
In the same line, it would be interesting to know if it does anything directly for the mast cell degranulation.
Yeah. That's something that we never do, and we always say that we're going to do. And I think it's like most of these salivary proteins, they have to do something with the mast cells, the granulation. Alvaro.
So I was wondering if part of the activity may be important inside the blood.
You're absolutely right. I think most of these proteins, the function is not in the biting side, it's in the midgut. That's where they're going to have the blood with all these nasty components for a long time. I think biting site is, if it's not something that affects really vasoconstriction or platelet aggregation, something that occurs in like about 5 seconds, everything else ends up in having the function in the midgut. So that's correct. So, well, with this conclude the session. The last speaker is not here. She didn't make it. So thanks for coming and enjoy the rest of the meeting. Thank you very much.